This short video is going to follow up the last one on titration and talk about indicators. In an acid-base titration, we have two ways that we can determine where we're at in the titration. Are we at the endpoint or are we not at the endpoint? One is by the use of a pH meter that might directly read out to us what the pH is. But the other one, and this is quite common, is to use something called an acid-base indicator. An indicator is something that visually shows whether a solution is predominantly acidic or predominantly basic by changing colors in different environments. What indicators are really is just weak acids that have a colored solution, one color or another, and their conjugate base have a different color. An example would be phenolphthalein, in which we see here the basic form is pink and the acidic form is colorless, doesn't interact with light to show any color. And what will happen here is the indicator, which I'll just call HIN for short, exists in equilibrium with H plus and its conjugate base. And so this is the pink and this is the colorless. And if we have a solution that has predominantly H plus in it, well, that's going to cause a shift of the reaction to the left. And so you would see the colorless HIN. If OH predominates, then that means OH is going to react with this H plus, and it's going to make water. But that removes H plus from the system, and the Le Chatelier response is to shift this system to the right, and that'll produce more IN minus, which is the pink colored stuff, and then you'd see that color. Here we see 4 beakers. Each one of them contains an ammonia solution. And ammonia is a weak base. So we should see the base color of any indicator that gets put in the beakers. I'm going to add indicators, a different one to each beaker. Into this beaker I'm putting methyl orange, and you can see that the base color is a kind of a yellowish orange color. Into this beaker I'm putting bromethymol blue, and the base color looks to be a sort of a blue color. I have that in the wrong spot on my paper, so I switched it. Into this beaker I'm putting thymol blue. Again, we see a blue color in the basic environment. And into the last beaker, I'm going to put an indicator called phenolphthalein. And we can see that in the basic environment, it's pink. After swirling, the colors are clear and continuous throughout. Now, to change the environment, I'm going to add an acid. And to this set of beakers, I'm going to add 3 molar hydrochloric acid, which should change the environment from basic to acidic. Into the bromethymol blue beaker, I'm adding HCl, and we see that the color change goes from blue to a sort of a pale yellow. In the video, it may look clear, colorless. Into the methyl orange beaker, I add the acid, and the color changes from yellow to a pink or orangish color. Into the phenolphthalein, I add the acid, and I change to a colorless solution. And the thymol blue changes from blue to a pink. Now we see the basic colors of all these 4 indicators. Now there's a slight problem that arises when we're titrating acids and bases. The equivalence point is the theoretical place where the reactants have just neutralized each other. And we have determined some things about the equivalence point in the last video when we were theoretically going through some titrations. The endpoint is where our indicator changes color. Ideally, we would like the endpoint, where the indicator shows the reaction is completed, to match the equivalence point. It's not going to in all cases, and we have to exercise some caution in selecting which indicator we use because of that. Now, for most indicators, about 1/10 of the indicator must have changed form before you start to see that the new color is showing up. So here's what that means. Since we've created a buffer system, the pH, when the color change first shows up, will be equal to the pKa of the indicator, which is sometimes called pKin, plus the base 10 logarithm of the basic form concentration divided by the acidic form of the indicator's concentration. Now let's say we're adding some base to a solution of acid. So I have HIN, it's in equilibrium with H plus, and IN 1 minus. when I first start to see a color change, if I started out with some amount of this, let's say 1, after 1/10 of that has formed, which would be 1/10 of 1, I would begin to see a color change. So the pH at the color change would be equal to pKn plus the base 10 logarithm of 1/10 over 1, or 1 over 10. Well, the base 10 log of 1/10 is equal to -1. So the pH, when I would first see the color change, is pKn minus 1. Let's say I were doing it the other way. Let's say I had a basic system and I was adding acid to it. Well, that would cause a shift to the left. And if I started out with a concentration of 1, or for my in, I would end up with a concentration of 1/10 for HIN during that shift at some point, and that's when I'd first notice that there was a color change happening. So in that case, the pH at that color change would be pKn plus the base 10 logarithm of 1 over 1/10. But that would be equal to pKn plus the base 10 logarithm of 10. Well, the log of 10 is 1, and so this would mean pH was equal to pKn plus 1. So for that reason, our indicator is going to go through its complete color change that's visible to human eyes between pKn minus 1 and pKn plus 1. So in order to make the endpoint and the equivalence point match as closely as possible, we should always select an indicator that has a pKn that's as close to the expected equivalence point pH as we possibly can. Now, for a strong acid versus strong base titration, lots of indicators will work. This is a very forgiving selection process, and here's why. For one thing, the pH at the equivalence point is equal to 7. The other thing that makes this easy is that there's a very broad range from about here to about here, where there's a dramatic change in pH with very little change in volume. Throughout this whole change here, it's actually narrower than that, it's about like that. This is the total change in volume of OH minus added in this strong base added to a strong acid solution. And so an indicator that changes at low pH with a pKn about here, like methyl red, would work. And phenolphthalein, which has a pKn that's about 9, would go through its entire color change also in this very limited amount of additional volume. And so you'd catch the equivalence point with either of these two indicators. For a weak acid and strong base titration, or conversely for a weak base versus strong acid titration, indicator selection is more critical for a couple of reasons. One, the equivalence point isn't at 7 anymore. The equivalence point is going to be above 7 for a weak acid versus strong base titration. It's going to be below 7 for a weak base versus strong acid titration. So like in this example where we're doing a weak acid versus a strong base, the shape of this curve makes it impossible to use methyl red effectively because we're going to see the color change start happening very early in the titration right here. And the complete color change will have occurred by the time we're at pH 6, but we are not going to actually reach the equivalence point until we're at over pH 8, you can see from this chart. So we would completely miss the equivalence point if we used methyl red. Using phenolphthalein would be better because the pH at the equivalence point is right there. We're going to see the color change happen between about here and here, and so we would actually catch that equivalence point at our end point. So it's important to select an indicator that has a pKn that's very close to the expected pH at the equivalence point.